The overall goal of this procedure is to quantify social motivation in laboratory mice. This is accomplished by first training each test mouse to associate lever pressing with a social reward. The second step is to test each mouse in a series of sessions in which the amount of effort required to obtain the social reward increases across trials. Next, each test mouse is trained to discriminate between two lever and reward contingencies. The final step is to test each mouse in a series of sessions in which they can choose between these contingencies, resulting in either a social or food reward. Ultimately, these operant social motivation paradigms allow investigators to quantify social motivation and compare this level to that of a food reward.
The main advantage of this technique over existing methods is that this paradigm requires progressively more effort to obtain a social reward than tasks simply measuring time spent in proximity to a social partner.
Demonstrating the procedure will be Brianna Lum, Megan Raymond, Amy Patterson, and Marianne Slamma, undergraduate student researchers in my laboratory.
To begin, set up center channel modular shuttle boxes for use in testing social behavior. Mount a wire grid to the face of a programmable guillotine door to separate the chambers and allow limited social contact between them. The door divides the box into two chambers. Designate one chamber.